please make sure that your iClickers are good, because now I'll start using them on a regular basis. And, sometimes it might just be like one question or, a couple of questions throughout the time we're here together in lecture. So but just have it ready. So per session, it's not going to be a huge thing per session, but I will be using it. So the next topic I wanted to get into is the cell structure and function. And this is chapter 3. And so with what you're going to be doing in labs, let's see, starting, what is today? Today's Friday. You did the simple stain, didn't you? Right? Hey, did your streak and spread plates, did those work? Mm-hmm. Yay. Okay, good. And so with the simple stain, can't remember which one you looked at, but basically the simple stain, we call it the simple stain because there's just one stain that you use. And most of the microbes that you're going to see in lab will be round or rod shaped. A few might look kind of comma shaped or bent a little bit, like a comma. We're trying to get one that has a spiral shape. You didn't work with that one, did you? We're having problems getting it to grow, and it's fussy. So, but we're trying to get you to look at a spiral shaped one in the near future here. So, but the vast majority of what you'll look at will be the coccus or the rod. They're not posted. All right, let me see if I can get in there. So the coccus shape, that's the fancy word to say round, like a sphere. And then the rod, we also say, they can be rod shaped. Okay, so now it should be visible. And so the rod shape used to be called bacillus shape. However, that got really confusing because there's a really famous genus of bacteria called bacillus. So it's like, hey, wait, are you talking about the genus or are you talking about the shape? And so everyone said, let's just go and with rod, stick with rod. So if you accidentally hear me say bacillus, and then you're like, what is she talking about? Big rod. Rod shaped, pocket shaped, spirillum shape are probably the most common, but you can see on this that there are other options. Often you're told in intro micro classes, look for symmetry under the microscope, and that's likely something, a microbe, whereas something asymmetrical is probably like, just some kind of artifact from the sample. Yes and no. Yeah, I can understand why they teach it that way, but sometimes you'll see things that don't have great symmetry, and it's still a microbe. So just be aware that there can be some odd looking microbes out there that aren't perfectly symmetrical, like a rod shape that you're seeing. And so, by the way, the ones that are stuck, or I think they're ones that live in environments where they have to attach or they get swept away. So things like rivers, streams, or pipes, things like that, where they have to really kind of latch on. And then filamentous bacteria, I work a lot. The Streptomyces bacteria I work with are filamentous, which is they're kind of a peculiar outlier type of microbe compared to the ones that you'll see in class. All right, so we need to think about the membrane of the microbe, and you've had this information before in intro biology because it's pretty much the same idea, that you have a phospholipid bilayer composed of phosphate group attached to a glycerol, and then you have two fatty acid chains with glycerol. So I'm going through this quickly because you should know this already. And then if you take some phospholipids and drop them into some water, they automatically will form this phospholipid bilayer configuration where the fatty acids tuck away from the water. They don't like to interact with water, so they tuck away. Have a gap between the two bilayers. Okay, so the phosphate glycerol groups love to interact with water, so they'll be on the outside like that. They're happy to, and so that allows for that spherical membrane to form in the presence of water. Okay, so this should be like hopefully activating parts of your brain that you've already solidified. So that's not it though. Yes, we have a phospholipid bilayer around the microbe, but they also will put their proteins in, just like we put, we have proteins embedded. And some of the proteins will go all the way through the whole membrane. Some are just on the surface, outside surface, inside surface, just depends. And when you look at the whole width for a microbe, it's about 6 to 8 nanometers wide, and lots of different proteins, and it depends on the microbe, what kind of proteins. And they say the membrane is somewhat fluid. That's how they describe membranes in general of any organism. They say somewhat fluid. And so the cell membrane, excuse me, of the microbe is really, really important, because not only is it that barrier and it's semi-selective or semi-permeable, so it lets certain things in and out, but you have to also appreciate that they don't like have a skeletal system or anything like that, so they have to really have ways to strengthen their cell membrane. And then also the fact they don't have organelles. We have to keep that in mind, because that impacts the cell membrane structure as well. So when you think about how to strengthen up the cell membrane, when you look at microbes, it depends on the microbe. If it's a fungus, for example, then they tend to have sterols in their cell membrane, and we have sterols in our cell membrane. We're eukaryotic as well. And so like, cholesterol, that's a sterol. So we do need cholesterol in our diet because it does help strengthen our cell membrane. Bacteria, the prokaryotic cell type though, those, especially, well, they studied it most heavily in the bacteria domain. They have hopanoids. Some people say hopanoids, I say hopanoids. That is their version. So they don't have sterols, they do hopanoids instead, but it's the same function. It's a way to strengthen up the cell membrane, but still allow for that semi-permeable nature. And so it's a nice, relatively easy thing to do. So here you're looking at cholesterol in the center, sterols on the far left. So the left and middle are what you see in eukaryotic organisms, and then hopanoids, you can see it's similar in structure, chemical structure. So what they do for like the hopanoids, it's those yellow excited chemicals here, right where the fatty acids are, and again, having that pattern just strengthens the membrane a bit. Okay. All right. So let me. Okay. So one other thing, really quickly, I just want to point out, we're going to pick this up on Monday. So sometimes details matter. So I'm going to be talking to you a little bit about the difference in the main archaea and the bacteria. One of the big differences is the bacteria have an ester and the archaea have ether linkage. And that sounds like a picky detail, but it makes a world of difference in terms of this ether bond is strong. And so that really helps the archaea adjust to very hostile environments, much more so than the ester-linked type microbe. Okay, we'll stop there. Have a good weekend, and I'll see you on Monday.